We'll go ahead and get started. So, thank you to everyone who's made it today on a Friday afternoon. We have the wonderful Kamal as our speaker today, who's the founder of the Imaging and Flow Cytometry Facility at MIPS, Monash Institute of Pharmaceutical Sciences in Melbourne. I guess everyone knows Cam as our Image J Fiji guru, but his skill set extends beyond that, and he's here to talk about. I'm not even sure what he's here to talk about. He assured me he has a series of stream of consciousness slides and he will talk about stuff. So I don't have much more to introduce beyond that. So I'll hand over to Cam to take it away.
Cool, thanks, Sam. Hi, everyone. Um, yeah, like Sam said, and because I told her, this is a stream of consciousness, bunch of different projects we've either worked on in the past, currently working on, halfway through, waiting for things to break, and more of just a shotgun of some of the different things we've used, um, and what we're doing. So, and open to any questions, criticisms, whatever of it as we go along. So I guess we'll start simply with what we're using for everything. So obviously, the vast bulk of our frameworks are all built in Fiji. We do everything in Fiji just because that's easier. I know how to code in that. I don't necessarily code well in it, but I can code in it. And so then we just make it do lots of other things. So generally, we're now using Elastic most of the time for anything that needs thresholds and everything. We don't, very few things ever use a threshold anymore. Mostly everything is a custom machine learning model done in Elastic. The advantage of that is that we can work something up pretty quickly, and then end users can retrain their models as they need to, if things don't work like they should, and then they can feed them back through the existing code. So that works pretty well. Stardust was another go-to we used to use a lot, but we are now at this point because Stardust itself is kind of falling over. So in everything I've got today, pretty much everything is still using Stardust because I haven't got around to retooling it yet. But moving forward, everything will just be a cell pose model for nuclei instead because Stardust is just, no longer really supported properly. And then we have cell pose as our main go-to for a lot of stuff. So, these two images here are the ones straight from Cellpose's website. This one down here is done by one of our PhD students, who um, who is looking at neurites, and this is purely just trained on Brightfoot images. And we've improved this even more now. We can actually measure branching and everything in these neurites growing in dishes, purely from a bright field image on a standard, wide field scope. So we don't have to worry about fluorescent stains, anything complicated. Just a bunch of GPU to train stuff. Um, so if I click, oh, that works well. Why are we not moving forward? Okay. So I was kind of thinking the slant for most of this is going to be about doing leveraging AI and everything that we're doing now. So the genesis of us doing AI came a long time ago. And so this is kind of one of the first movies that we worked on. So this is from Ben Croker when he was at the WeHi, and at the time I was next door at the Ludwig, and they were capturing all these images, and they just turned up one. I got an email one day saying, "Hey, we heard you do image analysis. We'll come bring the Sayos and the Vegemite, and we'll have a meeting." And I had no idea what they were talking about until years later when I ended up at WeHi and realized that everyone has Sayos and Vegemite for morning tea, and that made a bit more sense. So they turn up with these movies. These are mouse B cells that have been isolated. These live imaging experiments are done in 96 watt plates over 3 or 4 hour time periods. And they have Cell Tracker in green, they have Annexin in purple, and they have PI in red. And the whole point was to ping every cell and track it over time, not the specific cell, but just the population, and give an idea of apoptosis. And that was the first thing. So this is in PNAS in 2011. And then, there was evolutions of this over the time, and eventually got to, and we'd see data like this where we can see, differences in properium iodide intensity, so apoptosis. And this is always a good example to show for just why you should do live imaging, because if you did time zero and time, 500, the answer is there's no difference between these two. But if you do all the time points, there is quite a dramatic difference in what's going on. So then what happened is, Ben moved on from WeHi, ended up at, I think this was done when he was at Harvard at the Boston Children's Hospital. And these are now no longer nicely isolated mouse B cells, but these are human cells, and they tend to not behave right. So you end up with these clumps and mess. And so our traditional methods that we were using, which is just standard, watershed segmentation, falls over. Doesn't work very well at all. And around about this time is when things like Stardust came out. So straight away we transitioned to doing Stardist in this, and suddenly data that was unanalyzable in our old pipelines now works again. And so we can take this and do the same sort of stuff where you track every single cell. And once again, you can see this is cell tracker, annexin and PI, just different colors, and our little masks output. So this became a better way of doing things. Works really well on nice little round things. And so this then evolved into being able to put through various patient samples where these are children with Kawasaki disease that have been treated with intravenous IgG, and we can then track what happens, how their cells behave, and everything else. And this then led to sort of being able to develop a more elaborate way of looking at cell death instead of just saying it's an XM positive or it's PI positive and it's dead. You can start looking at necrotic cells and pyroptosis and a few other branches. So this became the sort of standard breakdown for a while where we could do this sort of stuff on cells, and you can look at the different populations you can see down here, and depending on the background of the cells and the treatments and what happens, you can see all the different effects over time in the shifts in the population. So once again, this is just a routine method now that is used by the lab all the time. Ben has since moved on to UCSF, but we still UCSF, UCSD, UCSD. And so we still collaborate on this, and there's various variations that happen, which is what this is. So we added net formation stuff now. This is where you got cells that dump DNA out. So now we've got an even more elaborate classification where we can give 6 different classes to the cell, depending on what is happening. And that's only from these, few dyes. It's not anything really complicated. It is just cell tracker, Hurst, PI, and annexin, just to be able to figure out what's happening with the various cell compartments. And we can do all this. Um, And once again, we get this, but we have all the different populations and stuff comes out. So that's, pretty good. So I guess if that was Genesis, then the next chapter has to be Exodus, which is also from the same group. So Ben, once he was at UCSD, kind of got known as the microscopy guy. So lots of people just come to him and get stuff done. And he kind of helps facilitate it. So this was I need to update this. This is actually now published in PNAS, if I remember. And this is some cells in a dish, obviously. But this is COVID research. So these are lung cells. So the nuclei are just the nuclei of the cells, and the red is COVID spike protein. So this is a standard what is it, FFU assay, so focus forming unit or colony forming unit, whatever standard virology assay for measuring titer of virus and how infective it is. And so the initial approach from Ben was, hey, look, we're doing this, we're doing it fluorescently. Can we just count the colonies and save us doing things manually? It's like, of course we can do that. That's not a problem. So we could take these and we can easily train an elastic model. Initially, this was a thresholdy based approach, and then the issue is depending on the strain and a few other things, the intensity of that spike protein is incredibly varied. So being able to um, have an adaptive thresholding system, so having machine learning from Elastic trained on everything, it worked really, really well. You can see there we can outline every single one of these little clusters quite easily. And this is once again, poor old Stardist. This is still working as long as someone doesn't update that version of Fiji, I assume. So this is just all the nuclei that are in the clusters versus what's not. And we can take all of these and we can do distance, Euclidean distance measurements between clusters. We can also do things like perimeter versus convex hull ratios, which gives you an idea of how convoluted these clusters are becoming. Are they getting really kind of wobbly and all over the place, or are they staying fairly round? And so this is ran over 96 well plates. The thing I haven't mentioned here is that all this also leverages Robert Huss's CLIJ plugins for Fiji to accelerate all the binary operations. It took it from 20 minutes a plate down to about 4 minutes by GPU accelerating all the binary functions, just because it takes a lot to do a full well under CPU. So that made a huge difference to this as well. And so this is what Ben originally wanted, was give me a count of the focus forming units. And we actually I think we tried to call it mega FU COVID when we put it in the paper, but that got suggested we should change it. But if you go to the repository, we've got it, I think it's still called that. So they never updated it. And so we got this working. It was running for them. It's like, Ben, I haven't seen any data. Is everything working? He's going, yeah, we're just trying to figure out what to do with everything else. So this is now what came out. Well, it's not all of it. This is some of the parameters that they decided were interesting from each of the wells now. So then we can look at, you know, shapes and sizes and relative distributions and cluster sizes and everything else across all the different, in this case, these are different compounds to treat the cells to see what effect it has on COVID uptake and infection. Um, so then CellPose came out. CellPose was amazing. And we just keep doing everything in CellPose now. Because it's easy to train a model in, it's easy to get our users to train models in it. There's always some poor student that gets dragooned in to sit there on a computer and outline all the different things. Um, So this is like one of the first goes I had doing more complex stuff with was cell pose. So this is a nerve bundle of nerves. So each one of this is like a nerve bundle here, and every one of these is one nerve with a little sheath around it. And the question was, we want to measure the thickness of the sheath around all of these. And so the easiest way it ended up doing that was to train two cell pose models, one for the whole thing and one for just the middle. And then if you do simple math between them, you're left with rings really, really quite well. You can then take each of those rings and measure thicknesses without a problem across every single one of those nerves. And you can then start getting, reams of data as we always do. Um, so you can look at thickness, start ratioing things, whatever you want to do, looking at relationships for the data. Um, Unfortunately, that never went anywhere. I kind of got it all working, and then I never heard from them again. So if anyone ever wants to measure nerves in toluene blue stain stuff, we have a solution for you already. Um, this is our first go of doing stuff with just transmitted light. So we had someone from St. Vincent's who was doing imaging here. They still do. So these are just standard cells here, and they've gone and stained up a bunch of endosomes or lysosomes in the cells. And they had the data go, oh, I want to count all of my lysosomes. It's like, well, what have you got? It's like, I got bright field and I've got my lysosomes. It's like, that's tricky. But it turns out with enough time and patience with CellPose, you can get it to recognize these bright field cells pretty well. And it does a surprisingly good job once you give it enough variance to look at it and train on. When I say enough, we probably gave it maybe 30 images of this sort, not like an obscene amount. It didn't take a huge amount of time to do. And then once again, small dots works great with Stardist. And in this case, we're doing a bit of a cheat. Stardist really likes to see nuclei taken on a 20 times objective. These are definitely not that. But if you blow the image up 4 times, it thinks they are nuclei taken on a 20 times objective, and it works fine. You don't have to worry about training a model. So that worked well. And then we can get up with things like this, where we ping every single cell. We can count how many endosomes or lysosomes, sorry, there are in it. No, endosomes. It is endosomes. There we go. And then you can look at things like if we give, DMSO, we actually have some sort of treatment that's going to affect the number of endosomes in the cell, and you can see that we actually can easily see population shift where we gain more endosomes and they become bigger and everything else works really well. And once again, this is all automatic feed through scripts. So then this was an interesting one. So we had a group that joined us during COVID now. So Rebecca Ritchie came here from the Alfred and bought her group and became our new head of department for the department I'm embedded in. And they work on, various cardiac things to do with diabetes and other metabolic diseases. And so Miles, who's was a postdoc and now co-lab head with Rebecca, sort of came to me in the early days and went, oh, you do image analysis stuff. Can you help us? It's like, oh, good. We can get you guys doing something really cool. So they have these H&E images. And I said, what do you want? You want to be able to like count the nuclei? You want to measure cell area? What do you want to do? He's like, oh, no, no, no. Just when I was at the Alfred, we used to have like an acetate sheet I could put over my monitor and it had a grid drawn on it. And we'd use that to help count the cells. Can you make Fiji draw a grid on my image for me? Well, the answer to that is yes, and that's what we kind of did for a bit. Um, that's what they were doing, because what would happen is you take your grid. For those not familiar with this as a technique, you have your grid, and then you pick a cell that crosses a grid crossing point, has to have a nucleus in it, and then you trace it by hand and you add that to the ROI manager. And then you go find another cell, and you do that for about 100 cells, so you get a representative of what's going on in the section. And then you can measure length and breadth and area of all the cells and figure out what's going on. Um, so after much convincing that we should try something else, uh, we got a cell pose model trained up, and it can extract all the cells without a problem. That was a bit of a more challenging training set because something like H&E staining is a bit variant sometimes, and there's some other weirdness going on. So we at least could get something that pinged most of the cardiomyocytes without too much of a problem. Um, and we've also set a bunch of rules now that it's got to have a nucleus in it and only one. If it's binucleate or something else, we ignore it. So something like this cell here that doesn't have a nucleus wouldn't be measured because we're not sure what's going on. And so then, as long as it had a nucleus, we're pretty sure we've got a cross section through a cell and it's only one cell. And so now they used to do, between 100 and 200 cells, and it would take them weeks to get through a data set. And now we do thousands, I think, on average. I think it's now about 5,000 cells. A heart section get counted, and it takes about 5 minutes per image. And they're now finally getting through their backlog because what was happening is because it was manual, any new student or staff member that joined the lab was like, oh, you need to do this. And they'd give them, a bunch of images to outline. They'd eventually go, screw this, I'm not doing it anymore. And once they stopped getting new people in the lab, they stopped analyzing data because no one wanted to outline cells anymore. So we've at least got rid of that problem. Um, and this works pretty robustly now. Some weirdness happened with Monash histology going to a newer perio scanner, and all the color profiles were different, which was fun. We had to retrain a few things, but otherwise it works pretty robustly. And we even have some variations of this now where we can look at, left and right ventricle stuff in sections and stratify it a bit more than just looking at everything. The other one that came out of from that group as well was looking at kidneys. So we had a go at this a while ago using Elastic, and we could kind of get it to work really, really well on one image, and we could not get it to apply across a decent proportion of images. So it kind of just fell onto the back burner for a while, and then once again we went, hang on, well, cell pose is probably pretty good. So for those not familiar, these are the sort of filtering structures inside the kidney. You can kind of vaguely see them all here. So this is the glomeruli, and we want to measure what's happening with these because in diabetic models you end up with less of them. They get a bit sick and funny, and we want to be able to measure them. So the trick for that was we can get cell pose to recognize them, but you take these images and scale them right down and filter them a bit, and these essentially get looking like a single cell. And so we train it to recognize the little fuzzy shrunk down ones and then blow them back up and fit the masks back on the image. So there we go. We can count glomeruli. And as what happens with any of these projects, there's always capability creep, and it's usually from me saying, "What about this?" So we added a few more things. We went, okay, well, once we can pull out each glomeruli, we might as well train an elastic model to pick the different tissue types so we can measure the actual glomeruli itself and the Bowman's capsule around it and other bits and pieces, infiltrates of immune cells, whatever. And once again, Stardist, which boo, going away. We can also give you an idea of cell density within a glomeruli because you can have ones that are the same size, but some of the cells have died off, so other ones have compensated to fill up the space. And of course, you do that across every single glomeruli in a kidney. And you end up with a thing like this. So that's everyone's extracted out and put into a little grid just to show what it looks like. Which I coined as a glomtage. And the lab actually uses the word glomtage, so I'm very happy it worked. It stuck. They refer to it as that now. And this is just the same thing with the Stardist nuclei extracted out. And then once again, we get a whole bunch of different data. So this is the first data set I ran through it when I was working it up, and I went, oh, it's not very exciting. It hasn't really measured anything. It all looks like it's counting everything it should, but the data looks pretty beige. And then afterwards I went, oh yeah, that experiment never worked. We should have given you one that did work. Like, well, that would have been good to know. But one advantage with this and the heart one is because they've kind of already done this manually for years before, we have a lot of ground truth to go back to. We can actually run it through these systems and go, do we get the same answer you got? And if we don't, is it because our model's bad or strangely enough, outlining only 50 cells in a heart full of 10,000 isn't representative of what's going on. So we can sort of bounce back and forth and figure out what's going on. So it's working pretty well. This is once again now a routine sort of script that's ran. Um, we go to things a bit more complex and into fluorescence, which always works so much better. So this is work from a PhD student here called Annabelle. So the actual final version of this now is called Synapse, I think is what she's named it. Um, so this is a tumor, a breast tumor in a mouse. And these tumor cells have been transfected with a system called LEGO, which stands for lentivirus gene ontology. The first time I ever worked with Lego was actually with a Danish postdoc, which was kind of funny that the Danish person was working on Lego. Um, and so what this does is you get your cells, you hit them with these reagents, well, these constructs, and a cell will either take up nothing and you won't see it at all, it won't be fluorescent, or it'll take up a blue one. So there is what have we got? BFP, GFP, YFP, M cherry, and Katushka. So we have blue, green, yellow, red, and a far red protein. So a cell can take up any one of those, or none of them, or any combination thereof, and at kind of different expression levels. So you end up with potentially 32 different combinations of cells. And the reason for doing that is that you go, well, in my tumor, everything's sort of a homogeneous mash. And we can give them these IDs. So, cell one is a blue, red, and a far red one, and it gets a unique barcode assigned to it for that combination. Um, and the reason for doing that is that you take your tumor, but then any of these cells, so you can see there's a big clump of yellow cells here. If anything metastasizes out, it's usually going to come from one cell, and you'll know what clonal cell that came from because it'll be a yellow one or a blue one or a blue and green one or whatever. And I guess what we should say is before we get to this point here, we've single cell sorted and clonally expanded every one of these combinations. So we have 34 different cells. We can make up a combination of, 100,000 of each cell and inject that into a tumor and know that proportions are even. And also because we have the parental cells, we can always go back genetically and see what's different about them when we only ever see green cells metastasizing. Why don't all the other ones metastasize? What's special about them? So we can go back and do that sort of checking. So this is once again the old cell pose. We train it to pick all the cells in a tumor. Um, and you can see there his different levels of expression, and we can see that it's outlining cells quite nicely without a problem. So we know in the tumor, then we can already straight away go away in each channel and figure out our cutoffs for each of the different colors, essentially treat it like a FACS experiment, and work out our barcodes we have. And we can look at distributions and everything else, which is fine. But then you get to do the fun bit where there's also, it's a bit hard to see, but there are little white bits around here and a few other white flecks. So this lab's focus is on neural signaling in cancer. So these are actually nerve fibers stained up here. So part of this is all to then look at what cell type or what barcode is next to a nerve, and does that same cell type then appear in subsequent metastasis. So this is easy to measure distances between, that's what this is talking about, using distance maps from the nerves to figure out where a given cell is. And then this is what the metastasis look like. This is a lung section. And so you can see instead of it being that, nice confetti look, every single kind of metastasis is a unique color or combination of colors. So each one of these has sprung up from one cell that's embedded into the lung at some point and grown out. And what we find is there is certain clones of these initial barcodings we've done that are definitely more likely to ever become a metastasis, and some never do. So there is definitely, even though they were from originally the same parental source, there is something unique about those cells that makes them being able to go and metastasize where other ones don't. And part of that could be location, although if it was purely location, then we'd see always a different mix of clones in each different mouse. We always see the same clones metastasize in the mice. So there is something very unique about those specific clones. And you have a look in here, for example, you end up with a bit of a mess sometimes where this is probably, all this yellow one was one, but then there's like a little purple one that's come in and a peachy colored one over here that's blended in a little bit. So it's not always as clean cut, but it's kind of there. And this is a blood vessel. Here you can see there's a bit of nerve all the way around. So they follow, usually come in via blood vessels, but some of them hit a blood vessel, get out, and then go way far away from one as well. And so the other step we do with this is that's this thing. So we assign each of these metastasis a unique cluster ID, and then we can start saying, does cluster one equal whatever barcode? That's obviously not cells out there. And once we've also got these clusters, we can start looking at In this case, this is distance of each cluster from a nerve bundle or a blood vessel. These are the cell distributions within each of these clusters. So we end up with a huge amount of data pulled out from this. So Annie's become a bit of an R wizard now and has done a lot of data mining with this and doing UMAP and principal component analysis of these and figuring out what's going on. The end result is it's not as exciting as we thought, but it's okay. We have a cool pipeline now anyway, if nothing else. Um, so leaping backwards now, talking still Solpos, but kind of making it do things that we didn't think would actually work very well at all. So this is back to Ben. And this is once again where he's got stuck in doing imaging stuff for people in weird situations. So this is currently now under review at Nature Comms. I think it's at Nature Comms. And in this case, this is autofluorescence just taken on an IncuSight, one of the IncuSight imager systems. And these bigger white blobs are these coccidioides spherules. So this is the fungus that produces river valley fever in people in like the lower US states and Mexico. And the problem is that once that gets into your lungs, you really can't do much about it. It's there and it just grows and you end up quite sick. And the other little fainter ones are they've gone and put a bunch of neutrophils or macrophages on to see if they can get some sort of response and start killing these things off before they grow too big. And so the initial pass was, can we count these and measure them? But also, what do we do when it's contaminated, well, not contaminated, but when it's been spiked with a bunch of this stuff? Once again, Cellpose does an amazingly good job. We train two different models, the green one and the red one, train it for the spherules, train it for the neutrophils, and then you can go and count and do all the other same stuff again. And this doesn't require any staining. This is just shove these things in an IncuSight over time. So these are all time course experiments as well. And you just use the autofluorescent channel. I think it was off the red. I can't remember. But it works surprisingly well. And here it is as a whole well, and you can see we can pull out these very easily. Even when they're contaminated, we can pull them out. Or not contaminated when they've got other things in there. Um, this was a little bit of a challenge. It's a bit hard to see here. I should have maybe done a zoom in. But as you go around the outside of the well, the things invert. So what used to be a bright spheral now becomes a dark spheral on a bright background, just because of the optics of trying to image a whole well on an IncuSight system. But we could give it examples of everything, and it eventually learned enough. So once again, it worked really well here, and then we'd always miss everything around here, and it became a bit of a trial and error of adding more stuff. But end result is, well, now we're being transcribed as well. Okay. Do I say okay to that? Okay. Thank you. And so we can get, area of the spherules was one of the easy measurements to get. This is actually a figure from the paper now where we can actually look at drug additions and we can see the number of spherules go up and the size of them change. And once again, this is all 96 well plate high throughput. These are all imaged in PC3 as well because it's an effective agent. So and an IncuSight system locked away. So nice and easy to get data out. The last step for that only sort of happened a couple of weeks ago, is this is what those spherules look like when they get into your tissue. So this is actually a tissue section, and each one of these is one of those spherules that has actually expanded out and just blown the crap out of the lung tissue and stuff that it's in. What they want to be able to do is be able to actually count these and measure them. And they'd worked up something, I think they were using QPath, and they could kind of get it to maybe pick up a little chunk, but not the whole thing. And obviously it doesn't work when you go around here and variations of not picking it up or only picking up a little bit. So once again, train cell pose enough and you can pick them all up and they thinks they're cells. And then we can go further from that. So now this thing has been labeled fungal finder. We were going to try and call it fungi finder, as in FUNGUY, but once again wasn't allowed. And so now this is going to be called lungal finder, to go looking for lung things. And it's going to have the same sort of, oh crap, go back. No. Love it. There we go. Once again, the usual, capability creep. This is just using a marker for the sort of outer wall of these spherules and Dappy or Herxed. But there is a whole bunch of stains for all the different immunocells in here as well. This is done on the Orion platform, which is like the Acquia platforms and all this sort of stuff where you have many, many color cell stains. So now we're going to go through and mine it all out for what are the cell types and what are they related to and everything else will be the next job for this one. Once again, lots and lots of data. But there is actually now a master's student in Ben's lab who's going to take over some of this, which will be nice. Um, and then for people that don't like imaging, we have answers for you too. This is not facts, this is an image. But this is some, in this case, this is human intestine. We can see a little crypts and we can see a bunch of cells stained up, and we have some, we split this apart. This is a 7 color fluorescent panel. Well, this is technically not, this is second harmonic collagen. So 6 fluorescents and a collagen. And we want to be able to deal with this sort of large data. And so we can, pull out all our cells. We can use elastic models to pull out, in this case, this is PGP for nerves. This is pulling out the epithelial layer of the colonic crypt. And then we want to look at all these immune populations. Sorry, there's more to go. Distance mapping as well. And so the problem you end up with is you get every single cell, we're going to know what marker it's carrying, its distance from the epithelium, its distance from a nerve, its distance from the edge of the tissue, its relative distance to other things. And so you know a bunch of parameters, and it becomes very difficult to work with. Now you can get into R and start doing clustering, or you trick FlowJo into thinking it's a flow data, which is pretty easy to do. So they have if you want to do this yourself, you Google FlowJo roll your own, and there is a little blog entry and a little Java tool to take a Excel file and turn it into flow data. So that's what this is showing here. In this case, this is control tissue, showing in this case distance of a cell to PGP, so to a nerve, and distance from epithelium. So we'd have 2 different clusters of cells that either live close to nerves or live far away from nerves. Great. This is a human disease called Hirschsprung disease, which affects children. So control isn't really control. They don't tend to cut the intestine out of a child unless they really have to. It's just that this isn't Hirschsprung. That's about the best we can say. It's control. And in this case, what happens with Hirschsprung is their colon doesn't contract. And so the cure or whatever is surgical resection. They just take out the chunk of the colon. It isn't working. And we get that to have a look at. So there's a section of it that doesn't have any nerves in it, like it's aganglionic. And then there is other sections that are ganglionic. So this is essentially normal-ish tissue. This is very diseased tissue, and this is control in this case. So we can start looking at various different things. So like CD45 cells tend to be close to the epithelium. In our aganglionic one, you notice we kind of lose a bit of a population-ish, but it comes back in the ganglionic. So all this kind of leads to this idea that only parts of the tissue are actually affected. Other bits behave normally, as normally as it can. But this is done from numerous sections, hundreds of thousands of cells counted and fed through, flow software to make it fooled into thinking it was going to work. Once again, like I said, this is a stream of consciousness. This is leaping around everywhere. We're back to nerves. In this case, a couple of guys from University College London who are part of our Farm Alliance network here at MIPS. And they grow these nerves in a dish and they want to measure neurites. And there is a plugin called Neuron J, which involves you tracing a lot of these. And that's all it does is measure lengths and maybe a bit of basic branching. And so we thought, well, we can work something up with this. And weirdly, Elastic alone can actually differentiate all these different bits. And maybe people smarter than me will understand why, but I think it has to do with the way the filtering works. If I actually trace along one of these neurites when I train, it then obviously builds filter shapes around that tracing. And it then pulls out long, skinny things pretty well. We train it for the blobs and we train it for the cells. And then we can easily go through and pull out all the neurites. At the moment, there's a few red ones because we set a threshold cutoff of it had to be at least 10 micron long to be counted. We're still figuring out what the best version of that is. And you can see that they're also branched, so we can run this through branching analysis to tell you are the neurites just getting longer or are they becoming more complex and more convoluted. The only thing we can't get at the moment from this easily is a per cell information. And the other issue with any of these neurites, even when they're doing it by hand, like for example, this one here, is it from that cell to here, or is it from that cell to there, or do they join and fuse in the middle? We don't know. So we can just get a general idea of what's happening in treatments and different conditions, but per cell becomes a bit tricky. Um, and then lastly, because we here at MIPS we have a bunch of chemists that do things. So this was a one of these other things that, like, I assume we can do this, but let's figure out how. So to start off with, here is this is a probe that binds amyloid protein, so things involved in Alzheimer's and Parkinson's and that sort of stuff. And so these guys have developed these probes that will bind and will fluoresce. And so this is just some aggregation over time over, it's a 24-hour run. And this is done in like a 96 well plate. So we're only going to look at one well, but this is happening across a massive data set. And so over time, it accumulates up. And as the proteins aggregate, this probe binds them and lights up. And you can see it doing that there. So that's just a single Z plane over time. If I pick a given time point, these are actually 3D little things that are crashing out a solution. So this is just the intensity of them. They're not very bright, but they do work. And so we can see that. So we've got time, 3D, 96 well plate. And then this horrible looking image here is the fluorescent lifetime. These aren't bright. They don't throw a lot of photons, and we're going pretty fast so we can get through the plate. But there is an increase in lifetime. Or you can measure a lifetime. Once again, it's in Z as well. So we can obviously just, our old friend Elastic, we can train up some masks and we can extract each of those blobs. This is once again a single plane over time, or in Z. Great. So then we can use those to clean up all the data and just look at just the bit that actually has stuff in it and look at its lifetimes. Once again in Z. Trying to make sure I've done this right. Good. It does go the way I thought it would. So if I just take that blob there at the moment, and if we look at the mean lifetime, in this case, over time, you can see that it goes from nothing because there's nothing there, but it goes up. And then eventually it stabilizes here at about, just over 3 nanoseconds. But over depth, if I just do a depth, a Z scan over depth, it changes across the depth of the thing. It also, it's a bit hard to see because it's noisy and horrible, but it is also different across the width of the thing. So we have a 3D blob that has a lifetime probe in it that changes lifetime as these things aggregate and also at different layers within the blob, which are not nice round spheres, right? So we can easily give everything an ID and we can measure it, but then this is time at the moment. So this is a standard Euclidean distance across the blob, and you can see, well, there's the outside and there's the inside in distances. Great. But these are 3D, so we have to do 3D Euclidean distance, which you can do. It works. So that's the same sort of thing, but going from every dimension in your blob. We can then assign that via layers to the lifetime, and we can then measure the lifetime of the blob at different 3D layers across time. For every blob over all the times in a 96 well plate, and you end up with an Excel sheet that is unopenable. So now we log everything out as a single CSV file for each well. And poor Kai has had to, one, figure out how to get all this data to make sense, and two, how to tell anyone about it. So that'll definitely earn him his PhD, if nothing else, other than just building the probes. But it has worked surprisingly well. It's incredibly complex data, but 3D over time done purely in something like Fiji is interesting, at least, if nothing else, instead of having to do it through Imaris or anything else that costs money. Yeah. So that's kind of that. So it's a bit funny in the compression, but you can kind of see the layers going through here. It is just rings. So all we've basically done is taken these Euclidean maps, set the intensity for the first ring, put it down onto the lifetime image, measured the average lifetime, drawn that out into a mask, and then just done that for all the different layers in all directions. It's actually doesn't take too long to run. I'm surprised. Once it's done its elastic thing, everything else is pretty quick. And that is the end of that.